Right now, at the bottom of the Mariana Trench, something is eating, not surviving, not clinging to existence at the edge of the possible, eating actively, efficiently, with a biology so precisely engineered for this environment that it makes the pressure and the cold and the darkness irrelevant. The creatures that live at the deepest point on Earth are not the desperate remnants of life pushed to its limits. They are specialists. They are optimized. They are, in the most rigorous biological sense, exactly where they are supposed to be. And what they are, and how they work, and what their existence means for everything we thought we understood about where life can go, that is what this story is about. If you enjoy drifting into the deep ocean and discovering what lies beneath, please consider liking and subscribing. It truly helps more than you know. Are you ready? Let's begin. The hadal snailfish, Pseudoleparus swire, and its relatives does not look like a predator. It does not look, in any conventional sense, like an animal that has conquered the most extreme physical environment on Earth. It is small, typically less than 30 centimeters in length. It is pale, almost translucent, with a body that appears too soft and too fragile for water that presses inward at more than 1,000 atmospheres. It has no swim bladder, the gas-filled organ that most fish use to control their buoyancy, because at those pressures, a gas-filled bladder would be compressed into a point of no functional value in milliseconds. It has large, wing-like pectoral fins that it uses to maneuver through the water, and a body plan that, in the darkness of the hadal zone, makes it look less like a fish and more like a pale ghost drifting through the sediment. It is, by any surface standard of what an apex predator looks like, completely unimpressive. And yet it is the deepest living vertebrate known to science. Filmed at 8,336 meters in the Izu Ogasawara Trench in 2023, the deepest confirmed fish observation ever recorded. It was not struggling. It was not barely alive. It was swimming with the fluid efficiency of an animal completely at home in its environment, foraging over the sediment with the unhurried competence of a predator that has no natural enemies at that depth and no reason to rush. It has been found in stomach content analysis to prey primarily on crustaceans, amphipods, and small shrimps, which it locates through chemosensory detection and lateral line mechanosensory in conditions where vision is essentially useless. The snailfish does not need to see its prey. It can smell the chemical gradients diffusing from a food source at distances of many meters, and it can detect the pressure waves generated by the movement of small crustaceans through the water. In a world without light, it has made light irrelevant. The molecular mechanism that allows the snailfish to function at these depths, the mechanism that allows any fish to function at depths approaching the theoretical vertebrate limit, is trimethylamine N-oxide, TMAO. This molecule is known to surface biology primarily as the chemical responsible for the fishy smell of seafood. It is produced when fish tissue decomposes, and its odor is the reliable signal that the fish in question has been dead long enough for its tissue chemistry to have shifted. In living fish, TMAO has a completely different function. It is a piezolyte, a small organic molecule that counteracts the destabilizing effects of pressure on protein structure. Proteins, the molecular machines that run every biological process in every living cell, are held together by weak chemical interactions that are sensitive to the thermodynamic changes induced by pressure. At high pressure, these interactions are disrupted and proteins unfold, losing the three-dimensional structure that makes them functional. TMAO counteracts this by modifying the thermodynamic properties of the water surrounding the proteins, shifting the equilibrium between folded and unfolded states back toward the folded functional configuration. The protein remains intact. The enzyme continues to catalyze its reaction. The cell continues to function. The relationship between TMAO concentration and depth in fish is remarkably consistent across species and ocean basins. Shallow water fish have low TMAO concentrations. As depth increases across fish lineages adapted to progressively deeper environments, TMAO concentration increases proportionally. The relationship is linear and predictive. Measure the TMAO concentration in a fish's muscle tissue, and you can estimate the depth at which it lives with reasonable accuracy. This linearity has a consequence that sets a hard biological ceiling on vertebrate depth tolerance. TMAO itself, at sufficiently high concentrations, becomes toxic to cells. It disrupts cellular processes in ways that are distinct from its pressure stabilizing function and that cannot be compensated by further increases in concentration. The calculations suggest that the maximum TMAO concentration compatible with cell survival corresponds to a depth of approximately 8,200 meters. Below that depth, the TMAO required to stabilize proteins against a prevailing pressure would itself be lethal. Fish cannot go deeper. Not because no fish has tried or because no fish has evolved in that direction, but because the chemistry of pressure stabilization has a built-in limit that the laws of biochemistry do not permit to be exceeded. The snailfish filmed at 8,336 meters is operating beyond what the original calculations predicted, and its existence there has prompted revision of the theoretical limit upward to somewhere between 8,200 and 8,500 meters, depending on the assumptions built into the model. Its TMAO concentrations are among the highest measured in any vertebrate. Its proteins are maximally stabilized. It is, in a biochemical sense, at or near the absolute limit of how deep a fish can go. Below it, in the remaining kilometers of the Mariana Trench down to the Challenger Deep at 10,935 meters, there are no fish. Not because the food is absent, the amphipods that the snailfish eats are present in far greater abundances below the snailfish depth limit, but because the vertebrate body plan, with its requirement for proteins that must fold correctly to function, hits a wall that the chemistry of TMAO cannot push through. The transition from fish fauna to invertebrate-only fauna at around 8,200 meters is one of the most precisely mechanistically understood depth transitions in deep-sea biology. It is a limit set not by evolution, but by physics and chemistry, by the thermodynamics of molecular structure under extreme pressure. What lives below the snailfish is stranger and in some ways more impressive, precisely because it has solved the pressure problem through entirely different biological strategies. The amphipods, small crustaceans of the order Amphipoda, primarily of the genus Hirondelia in the Mariana Trench, are found in extraordinary densities at the bottom of the Challenger Deep. When researchers deploy baited camera systems to the trench floor, the amphipods appear within minutes of the bait arriving, swarming over it in numbers that can reach tens of thousands of individuals in a single deployment. They are not struggling to survive, they are thriving. Their solution to the pressure problem is the same one used by the snailfish: elevated TMAO concentrations, modified membrane lipid compositions, but their body plan imposes fewer constraints. Crustaceans have no internal gas spaces that pressure can collapse. Their exoskeleton is rigid but not pressurized. Their gills extract dissolved oxygen from the water without requiring the maintenance of a gas-filled respiratory organ. They are architecturally better suited to high pressure than vertebrates are, and this architectural advantage is reflected in their ability to colonize depths that fish cannot reach. The amphipods of the Mariana Trench are not a single ecological type. They occupy multiple functional roles in the hadal food web. Some species are primarily scavengers. Drawn to large food falls the carcasses of whales, fish, and other organisms that die in shallower water and sink to the trench floor. These species have highly developed chemosensory systems that allow them to detect the chemical signatures of decomposing organic matter at great distances, and they can arrive at a food fall within minutes of its descent, consuming it with an efficiency that leaves almost nothing behind. Other species are predators, hunting smaller invertebrates through the sediment with a combination of mechanosensory detection and direct contact. Still others appear to feed primarily on the microbial biofilms that coat the sediment surface and the rock faces of the trench walls. The hadal amphipod community is not a single ecological actor, but a diverse guild of organisms occupying different parts of the available niche space, interacting with each other and with the other components of the hadal food web in ways that are still being mapped. The chemical ecology of amphipod aggregation at food falls is one of the more spectacular phenomena in hadal biology. The speed with which large numbers of amphipods can locate and converge on a food source in complete darkness, at temperatures just above freezing, across distances of hundreds of meters, is a demonstration of chemosensory capability that exceeds what most surface ecologists would expect from small crustaceans. The antennae of hadal amphipods bear chemoreceptor cells that are structurally modified compared to those of their shallow water relatives. Larger receptor surface areas, higher receptor cell densities, and molecular receptor configurations that appear to be tuned to the specific chemical compounds associated with decomposing marine organisms. These modifications are not random variation. They are the product of millions of years of selection in a food-limited environment where the ability to locate sparse, unpredictably distributed food sources is the primary determinant of survival and reproductive success. The amphipod's antenna is a precision instrument built by evolution for a single purpose: finding food in the dark. The food web dynamics of the hadal zone are shaped by the episodic, unpredictable nature of food delivery from above. The constant background input of marine snow, the slow rain of organic particles from the surface ocean, provides a low but continuous energy flux to the trench floor. Superimposed on this background are pulse events, the arrival of large food falls, the turbidity currents triggered by earthquakes that sweep organic material from the trench walls to the floor, the seasonal variations in surface productivity that alter the composition and quantity of the marine snow reaching depth. The organisms of the hadal zone have evolved life history strategies that exploit both the background flux and the pulse events. The amphipod's capacity for rapid aggregation and high consumption rates at food falls reflects adaptation to pulse exploitation. Their baseline maintenance metabolisms, which must sustain them through the long intervals between pulses, are calibrated to the low energy conditions that prevail between events. They can eat enormous amounts rapidly when food is available, and they can wait, with metabolic rates slowed by the cold, for the next opportunity. This metabolic flexibility, the ability to shift rapidly between high activity exploitation of food pulses and low activity maintenance between pulses, is a feature shared by many hadal invertebrates and reflects a general principle of hadal ecology. The environment selects for opportunism and patience simultaneously. An organism that cannot exploit food rapidly when it arrives will lose to competitors that can. An organism that cannot sustain itself through the long intervals between food arrivals will not survive long enough to exploit the next pulse. The successful hadal organism is one that can do both accelerate and wait, consume and conserve, respond and endure. The amphipods of the Mariana Trench have been doing this for long enough that the adaptations required for both modes are built into their biochemistry and behavior as fundamental features of what they are. The sea cucumbers holothurians that crawl across the sediment at the bottom of the Mariana Trench represent a different solution to the same set of problems. Where the amphipods are mobile, fast-responding, and capable of explosive aggregation, the holothurians are slow, continuous, and systematic. They move through the sediment at rates measured in centimeters per hour, ingesting it and extracting whatever organic matter it contains: microbial cells, degraded organic compounds, tiny invertebrates, the microscopic residues of marine snow that have passed through multiple rounds of biological processing before reaching their current position in the sediment. The holothurian's digestive system is adapted to process enormous volumes of sediment with high efficiency, extracting the dilute organic content that would be energetically worthless to a more selective feeder. Their contribution to the hadal ecosystem is the processing of the sediment itself, the constant biological reworking of the trench floor that cycles nutrients, oxygenates the near-surface sediment layer, and maintains the chemical conditions that support the microbial community on which the entire food web ultimately depends. The polychaete worms that burrow through the sediment perform a similar function at smaller scale and greater depth. Polychaete segmented worms, among the oldest and most diverse animal lineages on Earth, are found at hadal depths across multiple independent lineages, each of which colonized the hadal zone separately and developed pressure tolerance through its own evolutionary path. The hadal polychaetes have modified membrane lipid compositions and elevated TMAO concentrations that parallel the adaptations of the amphipods, but their ecology is distinct. They process the sediment from below, bioturbating the deeper layers and bringing buried organic material back within reach of surface-dwelling organisms. Their burrows also create pathways for oxygen penetration into sediment layers that would otherwise be anoxic, enabling microbial communities to function at depths within the sediment that would otherwise be chemically hostile. The polychaete worm at the bottom of the Mariana Trench is not a dramatic organism. It does not aggregate in spectacular swarms or exhibit chemosensory feats that astonish researchers. It burrows and processes and aerates continuously and unhurriedly, maintaining the physical and chemical infrastructure of the hadal ecosystem from the inside out. What all of these organisms, the snailfish at its TMAO limit, the swarming amphipods, the patient holothurians, the bioturbating polychaetes have in common is that they are not there despite the conditions of the hadal zone. They are there because of those conditions. The extreme pressure, the cold, the darkness, the food scarcity, these are not obstacles that deep-sea evolution has struggled to overcome. They are the selection pressures that have shaped the specific biochemistries, body plans, behaviors, and ecological roles of the organisms that inhabit the hadal zone. Remove the pressure, and the snailfish's TMAO concentrations would make it sick. Return the amphipods to surface conditions, and their membrane lipid compositions would make their cells dangerously fluid. The hadal organisms are not adapted to the deep in spite of its extremity. They are built for it, precisely and completely, in ways that make the surface world as hostile to them as the hadal zone is to us. This is the deepest truth about the creatures of the Mariana Trench. They are not visiting an extreme environment. They are at home in it. The environment that seems, from the perspective of surface biology, to be designed to destroy life, is from their perspective simply the world, the only world their lineages have known for millions of years. The world whose specific parameters are encoded in every molecule of their cells, and the diversity of solutions they have evolved to function in that world, the different molecular strategies, the different body plans. The different ecological roles points towards something that the next part of this story explores. Not just how these organisms survive, but what their survival implies about the range of conditions that life, anywhere, can occupy. The foraminifera at the bottom of the Challenger Deep deserve particular attention because they represent, in one organism, the most extreme extension of biological scale in the hadal zone. Foraminifera are single-celled organisms, not multicellular animals with specialized tissues, but single cells, managing all biological functions within the boundaries of one continuous cellular membrane. In the surface ocean and at shallow depths, foraminifera are typically microscopic, a millimeter or less in diameter, their shells constructed from calcium carbonate or agglutinated sediment particles. At the bottom of the Mariana Trench, researchers have found Xenophyophores giant foraminifera with shells that can exceed 20 centimeters in diameter. These are single cells the size of a human fist. They are among the largest single-celled organisms on Earth, and they live at the deepest point of the deepest trench, in conditions that would prevent the existence of most multicellular life. The xenophyophore success at hadal depths is partly a function of its architecture. A single cell has no gas spaces to compress, no tissue boundaries to maintain under pressure differential, no complex organ systems whose function depends on specific pressure-temperature conditions. The challenges of extreme pressure fall more heavily on the complexity of multicellular organization than on the fundamental chemistry of cellular life. The xenophyophore, by remaining a single cell, has sidestepped the structural vulnerabilities that make extreme depth so challenging for animals. What it has not sidestepped is a biochemical challenge. Its membranes and proteins face the same pressure as everything else in the trench, but the evolutionary solutions available to a single-celled organism, with its enormous population sizes and rapid generation times, are different from those available to a slowly reproducing animal. The xenophyophore at the bottom of the Challenger Deep is a reminder that the constraints on life at extreme depth are not universal. They are specific to particular types of biological organization, and the organisms that have escaped those constraints have done so by being built differently from the start. The microbial community at the bottom of the Mariana Trench is the foundation of everything, not metaphorically, literally. Every gram of organic matter that reaches the trench floor passes through microbial processing before it becomes available to the animals above in the food web. The amphipods eat organic matter that bacteria have partially degraded. The holothurians ingest sediment whose nutritional value is almost entirely determined by the microbial biomass it contains. The polychaetes aerate sediment layers whose chemical conditions are maintained by microbial metabolism, remove the microbes, and the hadal food web collapses entirely within days. They are not a component of the ecosystem. They are the ecosystem's engine. The bacteria and archaea of the Challenger Deep sediment are among the most pressure-adapted organisms known to science. A single cubic centimeter of hadal sediment contains hundreds of millions of microbial cells, a density that, given the extreme scarcity of organic input at those depths, seems paradoxical until the metabolic rates are considered. These microbes are extraordinarily slow. Their metabolisms operate at a fraction of the rate of surface microbes, constrained by the cold and the pressure and the chemical conditions of the hadal sediment. Some hadal bacteria have been estimated to divide once per 1,000 years or less, a generation time so long that the entire recorded history of human civilization represents fewer than 10 bacterial generations. They are not inactive. They are patient in a way that the human sense of time cannot easily accommodate. The archaea, a domain of life distinct from bacteria, often found in extreme environments, are particularly abundant in hadal sediment. Certain archaeal lineages specialize in anaerobic metabolism, breaking down organic compounds in the absence of oxygen through fermentation and other pathways that do not require the electron acceptors that aerobic organisms depend on. In the deeper layers of hadal sediment, where oxygen has been consumed by the surface microbial community and cannot be replenished by diffusion from the overlying water, archaea dominate. They process organic matter through metabolic pathways that have no analog in the biology of the sunlit world, pathways that operate under conditions of zero oxygen, extreme pressure, near freezing temperature, and organic carbon concentrations so low that surface microbiologists would classify the sediment as essentially sterile. The specific metabolic pathways used by hadal microbes have been characterized through metagenomic analysis, the sequencing of all DNA present in a sediment sample, which reveals the genetic toolkit available to the microbial community without requiring the cultivation of individual organisms. Hadal sediment metagenomes differ substantially from those of shallower marine sediments. They contain elevated proportions of genes encoding pressure-stable enzyme variants, genes for the biosynthesis of the unusual membrane lipids that maintain fluidity under extreme pressure, and genes for metabolic pathways that are rare or absent in shallow water microbial communities. They also contain a high proportion of genes whose functions are not yet known sequences that do not match anything in the existing databases of characterized genes from any environment. The hadal microbial community is not simply a pressure-adapted version of the shallow water microbial community. It contains biological capabilities that have not yet been documented anywhere else. The organic matter processing that drives hadal microbial metabolism is not uniform across the sediment profile. The surface layer of the sediment, the zone, in direct contact with the overlying water, is aerobic, oxygenated by the slow diffusion of dissolved oxygen from the bottom water. Here, aerobic bacteria dominate, using oxygen as their terminal electron acceptor in respiration and achieving the highest metabolic rates in the hadal sediment column. Below the aerobic zone, which may extend only a few centimeters depending on the oxygen consumption rate, conditions shift to anoxic. The transition from aerobic to anaerobic conditions is not a hard boundary, but a gradient tracked by the concentration of dissolved oxygen and by the shift in the dominant microbial community. Below the oxic-anoxic boundary, archaea and anaerobic bacteria take over, processing the organic matter that the aerobic community above has partially degraded, using sulfate, nitrate, and other oxidized compounds as electron acceptors in place of oxygen. This vertical stratification of microbial metabolism within the sediment is a microcosm of the vertical zonation of the entire water column above the progression from oxygen-rich surface waters through progressively more anoxic conditions with depth reflected at millimeter scale in the sediment of the trench floor. It creates a chemical gradient that drives diffusion of nutrients and metabolic products between layers, connecting the aerobic surface to the anaerobic depths and maintaining a continuous flux of chemical energy through the entire sediment column. The sediment of the Challenger Deep is not a passive accumulation of dead material. It is an active chemical reactor, processing organic matter through a layered community of microbes that together extract every available calorie from the material that reaches them. The rate at which organic carbon is mineralized, converted back to carbon dioxide by the hadal microbial community, has implications that extend beyond the trench itself. The deep ocean is one of the major long-term reservoirs of organic carbon on Earth, and the rate at which microbial communities in the deep sediment remineralize that carbon determines how long it remains sequestered and out of contact with the atmosphere. In the hadal zone, where organic input is concentrated by the geometry of the trench and where the cold slows microbial metabolism, carbon residence times are long. The carbon in hadal sediment may remain there for thousands of years before being remineralized. The hadal zone is, in this sense, a carbon sink of disproportionate significance relative to its area, a small fraction of the ocean floor by surface area, but a repository of organic carbon that persists on timescales far longer than the average for the deep sea. The discovery that hadal sediment contains measurable concentrations of human-derived chemical pollutants, polychlorinated biphenyls, persistent organic pollutants, and microplastics has revealed that the microbial community there is now processing material that has never previously existed in any natural environment. PCBs, manufactured primarily between the 1930s and 1970s, were designed to be chemically inert and resistant to degradation. They are, by design, difficult for microbes to break down. Yet hadal sediment contains PCB concentrations that, in some studies, exceed those found in the sediment of heavily industrialized coastal estuaries. The PCBs arrived via the biological pump accumulated in the tissues of surface organisms, concentrated through food chains, and eventually carried to the trench floor in the bodies of organisms that died and sank. The microbial community of the Challenger Deep is now confronted with compounds that its evolutionary history contains no preparation for processing. Whether it will develop the capacity to degrade them, whether evolutionary pressure over sufficient time will produce microbial strains capable of PCB mineralization, is unknown. The timescales of hadal microbial evolution are long. The persistence of PCBs in the environment is also long. Whether the two will converge before the compounds cause irreversible harm to the microbial community that underlies the entire hadal food web is a question that no one has yet answered, partly because the microbial community of the Challenger Deep has been studied in enough detail to ask the question only in the past decade. The microplastics that have been found in hadal sediment present a different kind of challenge. Unlike PCBs, which are dissolved compounds that microbes might in principle learn to metabolize, microplastics are physical particles, fragments of synthetic polymer ranging from a few micrometers to a few millimeters in size. They accumulate in the sediment as a physical addition to the mineral and organic matrix, changing the texture and porosity of the sediment in ways that affect the microbial community's access to oxygen, water, and organic substrates. They also carry surface-associated contaminants, the chemical additives incorporated into plastic during manufacturing, and the persistent organic pollutants that adsorb to plastic surfaces from the surrounding water during their long descent through the water column. The microplastic particle that reaches the Challenger Deep floor is not simply a piece of inert polymer. It is a chemical delivery vehicle carrying a payload of compounds that the hadal environment has never encountered and that the organisms there have no evolved capacity to handle. The virus community of the hadal zone is one of the least studied aspects of hadal microbiology and one of the most ecologically significant. Viruses are the most abundant biological entities in the ocean. Marine water contains on the order of 10 million viral particles per milliliter, and deep-sea sediment contains comparable densities. Most of these viruses are bacteriophages, viruses that infect bacteria and archaea. In the surface ocean, viral lysis, the destruction of bacterial cells by viral infection, kills roughly 20% of the bacterial community every day. releasing the organic contents of those cells back into the water where they are consumed by other bacteria. This viral shunt, as it is called, is a major pathway of carbon cycling in the surface ocean. At hadal depths, viral lysis rates appear to be lower than at the surface. The extreme conditions slow viral replication as surely as they slow everything else, but the viral community is still active, still infecting and lysing bacterial cells, still shunting carbon through pathways that would not otherwise exist. The viruses of the hadal zone are also, in many cases, genetically distinct from those of shallower environments, carrying genes that may encode novel biochemistries adapted to the extreme conditions of the deep trench. Every viral genome sequenced from hadal sediment has contained genes with no known function, dark matter, in the genetic library of the deep, pointing toward biological capabilities that science has not yet characterized. What the microbial and viral communities of the Challenger Deep collectively represent is a biological world of extraordinary antiquity and complexity, operating at the limit of what the laws of chemistry and physics permit, processing the chemical legacy of the surface world through metabolic pathways that took billions of years to evolve, and doing so now in the presence of compounds that no evolutionary history prepared them for. They are the oldest living community on the trench floor bacteria and archaea, whose lineages extend back further than any animal, whose biochemical innovations provided the foundation on which all subsequent life on Earth was built. And they are, simultaneously, the organisms most directly exposed to the consequences of what the surface world has done to the deep ocean in the past century. The bacteria in the Challenger Deep sediment are older than the Mariana Trench itself, descendants of lineages that were ancient before the Pacific Plate began its subduction. The PCBs in that sediment are younger than some people alive today. The collision of those two timescales, the deep time of microbial evolution and the shallow time of industrial pollution, is playing out at the bottom of the world, in the dark, without witnesses, in ways that will determine the health of the hadal ecosystem for longer than any human institution has yet existed. The microbial community will not disappear. Life at those depths has survived conditions far more severe than the arrival of synthetic polymers and chlorinated biphenyls. But the specific community that exists there now, the specific genetic diversity, the specific metabolic capabilities, the specific ecological relationships between microbes and their viral predators and their animal consumers is being altered by inputs that evolution has had no time to respond to. The hadal zone is not fragile in the way that a coral reef or a rainforest is fragile. It is ancient and resilient and capable of surviving changes that would destroy surface ecosystems. But resilience has limits, and the limits of hadal microbial resilience are not known because they have never been tested at the pace and scale at which they are currently being tested. The organisms that the next part of this story explores are the beneficiaries of everything the microbial community produces, the animals that sit above the microbes in the food web, consuming what the microbes make available, shaped by millions of years of evolution in a world where the microbes set the terms. The question of how microbial communities at hadal depths differ between trenches between the Mariana and the Kermadec, between the Tonga and the Puerto Rico, is one that metagenomic analysis is only beginning to answer. Early comparisons suggest that hadal microbial communities are not globally uniform. Each trench has its own characteristic microbial assemblage, reflecting the specific physical and chemical conditions of that trench, the temperature, the organic input rate, the sediment composition, the history of geological disturbance. The Mariana Trench microbial community is shaped by the specific organic input from the overlying Pacific Ocean, which differs in composition from the Atlantic input to the Puerto Rico Trench, or the Southern Ocean input to the South Sandwich Trench. These differences propagate up through the food web, influencing the chemistry of the organic matter available to the animals above, and potentially influencing the specific adaptations of those animals to their particular trench environment. The viral communities show even more trench-specific variation than the bacterial communities. Viruses evolve rapidly, and their genetic diversity tracks the genetic diversity of their hosts with high resolution. The viral metagenomes of different hadal trenches are, at the sequence level, largely distinct, suggesting that the viral communities, like the bacterial communities, have diverged significantly between trenches over the time that those trenches have been isolated from each other. This isolation is not complete. Deep ocean currents carry viral particles between trenches over long timescales, and occasional colonization events connect otherwise isolated communities. But the predominant evolutionary dynamic in hadal viral communities appears to be local adaptation, the accumulation of genetic changes that tune viral biology to the specific hosts and conditions of the trench in which they have been evolving. The implications of this trench-specific microbial diversity for the management and protection of hadal ecosystems are significant. If each trench contains a unique microbial and viral community and a distinct genetic library of biological capabilities found nowhere else, then the loss of any trench's community represents an irreplaceable loss of biological diversity. The organisms visible to cameras and nets, the amphipods, the holothurians, the snailfish, are the tip of an enormous microbial iceberg. Below them, in the sediment, is the majority of the trench's biological diversity, the majority of its ecological function, and the majority of its evolutionary history. Understanding and protecting hadal ecosystems requires understanding and protecting not just the visible animals, but the invisible microbial community that makes those animals possible. The rate at which new microbial lineages are being discovered in hadal sediment through metagenomic sequencing of samples collected by the small number of expeditions that have reached the Challenger Deep suggests that the true microbial diversity of the hadal zone has barely been sampled. Every expedition returns with sequences that match nothing in the existing databases. Every new trench sampled contains microbial lineages not found in previously sampled trenches. The microbial dark matter of the hadal zone, the genes and genomes encoding functions that science has not yet characterized, represents one of the largest unexplored frontiers in biology. What those unknown genes encode, what biochemical capabilities they represent, what metabolic pathways they enable, these are questions that will be answered only by the expeditions that have not yet been conducted, with the analytical tools that are only now becoming available, the zombie worm does not look like something that should exist. Osedax, the genus name, means bone eater. In Latin, is a polychaete worm, a member of the same broad family of segmented worms that burrows through the sediment of the hadal zone. But Osedax does not burrow through sediment. It burrows through bone. Specifically, the bones of whales that have sunk to the seafloor, coming to rest in the deep ocean, as what biologists call whale, falls the carcasses of the largest animals on Earth, slowly decomposing on the ocean bottom over years and decades, providing a concentrated resource of organic matter and inorganic minerals in an environment where both are ordinarily scarce. The Osedax worm was discovered in 2002 during a dive by the remotely operated vehicle Tiburón off the coast of California. When researchers noticed a whale skeleton at 2,891 meters covered in what appeared to be small red plumes, those plumes were the exposed gills of Osedax worms, hundreds of them, packed together so densely that they obscured the bone surface beneath. The worms had no mouth. They had no gut. They had no digestive system of any kind. And yet they were clearly alive, clearly metabolizing, clearly consuming something from the whalebone. What they were consuming, and how, turned out to be one of the more remarkable discoveries in the biology of the deep sea. Osedax worms penetrate whalebone using specialized root-like structures that branch and spread through the bone matrix, secreting acids that dissolve the mineral component of the bone and releasing the lipids, whale oil, locked within. The worms cannot digest these lipids themselves because they have no gut. Instead, their root tissues are packed with endosymbiotic bacteria, chemosynthetic organisms that live inside the worm's cells and use the lipids released from the bone as an energy source, producing organic compounds that the worm absorbs and uses for its own metabolism. The worm is not eating the whale. The bacteria inside the worm are eating the whale, and the worm is farming the bacteria, providing them with access to the bone's lipid reservoir in exchange for the nutrients they produce. The similarity to the hydrothermal vent tube worms, which also lack a gut and farm endosymbiotic chemosynthetic bacteria, is not coincidental. It is a demonstration of evolutionary convergence. Two different animal lineages facing the same basic challenge of extracting energy from a chemical source in the dark, arriving at the same basic solution through independent evolutionary paths. The ecological role of whale falls in the deep-sea ecosystem is disproportionate to the frequency with which they occur. A single blue whale carcass, the largest animal on Earth, potentially exceeding 150 tons, represents an enormous input of organic matter and inorganic minerals to the deep-sea floor at a single location. Estimates suggest that a large whale fall can support a distinctive community of organisms for 50 years or more, providing habitat and nutrition to species that are specifically adapted to exploit whalebone chemistry, and that would not survive in the absence of whale falls. These whale fall communities include not only Osedax worms, but specialist polychaetes, mollusks, crustaceans, and microbial communities that are found nowhere else in the deep ocean. They are, in effect, oases concentrated patches of biological productivity and diversity in an otherwise food-limited environment, connected to each other across the deep-sea floor by the movements of organisms that track whale carcasses as they become available. The discovery of osedacs has prompted a reassessment of deep-sea biogeography. Because whale-fall communities provide stable habitat for extended periods, and because the organisms that exploit them include species capable of dispersing between whale falls across large distances, the whale falls of the global deep ocean may function as stepping stones, a series of temporary habitats that allow deep-sea species to colonize new areas and maintain genetic connectivity between otherwise isolated populations. The geography of whale falls, which depends on where whales live and where they die, overlaps partially but not completely with the geography of hydrothermal vents and cold seeps, the other major oases of the deep sea. Together, these patchy, distributed habitats support a biogeography of the deep ocean that is more connected than the distances between them would suggest, linked by the dispersal of organisms through open water and by the occasional coincidence of organism and habitat that allows colonization of new sites. The osedac story also has a temporal dimension that reveals something important about the evolutionary history of the deep sea. Osedacs is highly diverse. More than 20 species have been described since the genus was discovered in 2002, and molecular analyses suggest that the true species diversity is substantially higher. The divergence times of the major osedac lineages, estimated from molecular clocks, suggest that the genus has been present in the deep ocean for at least 40 to 50 million years, predating the diversification of the large baleen whales, whose bones it primarily exploits today. This creates a puzzle. What were the ancestors of osedacs eating before large baleen whales became abundant? The molecular clock evidence suggests that osedacs evolved to exploit bone-embedded lipids long before the whale falls that currently sustain it became a major feature of the deep-sea environment. The probable answer is that ancestral osedacs exploited the bones of the marine reptiles ichthyosaurs, plesiosaurs, mosasaurs that dominated the Mesozoic ocean before the rise of modern whales. When those animals went extinct at the end of the Cretaceous, the osedac lineage survived by shifting to the bones of the archaic early whales that evolved during the Paleocene and Eocene. As modern large baleen whales evolved and their populations expanded during the Miocene, osedacs diversified explosively, tracking the expanding resource. The worm that looks like a novelty, a tube of tissue with no mouth, boring into bone, is in fact one of the oldest ecological specialists in the deep sea, a lineage that has been exploiting vertebrate skeletal lipids since before the Rocky Mountains were built. The cold seeps areas of the deep sea floor where hydrogen sulfide, methane, and other reduced chemical compounds seep upward through the sediment from geological sources below are another category of deep-sea oasis that supports communities distinct from both the surrounding deep-sea floor and hydrothermal vents. Cold seeps are not hot. The fluids that emerge from them are close to the ambient temperature of the surrounding deep water. But like hydrothermal vents, they provide a chemical energy source, the reduced compounds in the seeping fluids that supports chemosynthetic microbial communities, which in turn support complex animal assemblages. Cold seep communities include tube worms related to the hydrothermal vent Riftia, though belonging to different genera, dense beds of mussels and clams whose tissues contain endosymbiotic chemosynthetic bacteria, and a diverse fauna of crustaceans, echinoderms, and other invertebrates that exploit the bacterial production directly or through predation on the chemosynthesis-dependent organisms. Cold seeps are found in many deep ocean environments, including some locations within or adjacent to the Mariana Trench system, and their communities are thought to persist for thousands of years at individual sites, far longer than hydrothermal vent communities, which shift as the geological activity that drives them changes. The methane seeping from cold seep environments has a chemical fate that connects the deep-sea biology to the global carbon cycle in ways that are only beginning to be understood. Methane is a potent greenhouse gas, far more warming per molecule than carbon dioxide over short timescales, and the amount of methane stored in seafloor sediments as methane hydrates, ice-like structures in which methane molecules are trapped within a crystal lattice of water, is enormous. Estimates suggest that the seafloor methane hydrate reservoir contains more carbon than all the coal, oil, and gas reserves currently known. If this methane were released to the atmosphere, as might happen if deep-sea temperatures rise enough to destabilize the hydrate deposits, the greenhouse effect would be dramatic. The microbial communities at cold seeps are the primary biological barrier to this release. Anaerobic methane oxidizing archaea, working in partnership with sulfate-reducing bacteria, consume most of the methane that seeps upward through the sediment before it reaches the water column. They do this through a process called anaerobic oxidation of methane, coupling methane oxidation to sulfate reduction in a metabolic partnership that neither organism can perform alone. The efficiency of this microbial methane filter is high. Estimates suggest that it consumes more than 90% of the methane that would otherwise escape from marine sediments globally. The archaea at the cold seep that look, under a microscope, like nothing more than tiny spherical cells embedded in sediment, are performing one of the most consequential carbon cycling functions on the planet. The connection between cold seeps, methane hydrates, and the global carbon cycle is one of the reasons that deep-sea biology is not merely a matter of scientific curiosity about exotic organisms. The organisms of the deep sea are part of the Earth system. Their metabolic activities regulate the chemistry of the ocean and the atmosphere. Their response to changes in deep ocean temperature and chemistry will determine whether the enormous carbon reservoir locked in seafloor sediments remains locked or begins to leak. The organisms that the deep sea has evolved over billions of years to perform these regulatory functions, the methane oxidizing archaea, the sulfate-reducing bacteria, the chemosynthetic communities of cold seeps and hydrothermal vents, are not separable from the planetary processes they regulate. They are those processes, encoded in biological form, maintained by the same evolutionary pressures that have maintained every other feature of the living world. And the strange, improbable, fascinating animals that have evolved to exploit the chemical landscapes these microbes create, the osedacs boring through whalebone, the tube worms at the vents, the clam beds at the seeps, are not peripheral oddities. They are the visible manifestation of biological systems that connect the deepest ocean to the global climate through chains of metabolism and ecology that took billions of years to assemble and that science is only beginning to trace. The dispersal biology of deep-sea chemosynthetic organisms raises questions that the discovery of Osedax crystallized, but that apply equally to every organism dependent on patchy, distributed habitats in the deep ocean. How do organisms find new habitats when their current habitat disappears? How do populations maintain genetic connectivity across the vast distances of the deep ocean floor? How quickly can a newly formed whale fall, a newly opened hydrothermal vent, or a newly active cold seep be colonized by the specialist organisms that depend on those habitats? These questions are not merely academic. They determine the resilience of deep-sea communities to disturbance their ability to recover from the episodic catastrophes of turbidity currents, vent shutdowns, and sediment burial that regularly reshape the physical landscape of the deep ocean. The larvae of deep-sea invertebrates provide the primary mechanism of dispersal. Many hadal and deep-sea species reproduce by releasing larvae into the water column small, free-swimming developmental stages that can be carried by currents across distances of hundreds or thousands of kilometers before settling at new habitats. The duration of the larval stage and the sensitivity of larvae to deep-sea conditions during transit determine how far dispersal can occur. Some species have short-lived larvae that must settle quickly and therefore cannot disperse far. Others have long-lived larvae that can persist in the water column for months, potentially crossing entire ocean basins before finding a suitable habitat. The genetics of deep-sea populations, the patterns of relatedness between individuals from different locations, reflect the historical dispersal ability of each species and reveal which populations are connected and which are isolated. For Osedax specifically, genetic studies have revealed that populations from different ocean basins, the Pacific, the Atlantic, the Indian Ocean, are genetically distinct, consistent with limited dispersal across the deep ocean ridges and other barriers that separate the major ocean basins. Within ocean basins, however, populations show surprisingly high genetic connectivity, suggesting that larval dispersal regularly connects distant whalefall communities and maintains genetic exchange across distances of thousands of kilometers. The whale falls of the Pacific Ocean form, in this view, a network of temporary habitats connected by the dispersal of Osedax larvae through the deep water, a network whose geometry shifts over time as individual whale falls are colonized and then exhausted as new ones form, and as the populations that exploit them track the changing landscape of available resources. This network view of deep-sea habitat, patchy, dynamic, connected by dispersal, maintained over geological time by the continuous generation and exhaustion of individual habitat patches, applies not just to Osedax, but to most of the chemosynthesis-dependent communities of the deep ocean. The hydrothermal vent communities that inspired a reconsideration of where life can exist, first discovered in 1977 and now known from hundreds of sites on mid-ocean ridges around the world, are themselves patchy and temporary, each vent site active for decades to centuries before the geological activity that drives it shifts elsewhere, the community it supports dying or dispersing as the vent goes extinct. The communities that colonize new vent sites are assembled from the larvae that happen to be in the water column when the vent opened, drawn from the nearest active vents and from the deep water larval pool that connects them. Each new vent community is a unique assemblage, reflecting the specific colonization history of that site, the species pool available from adjacent vents, and the specific physical and chemical conditions of the new vent environment. No two vent communities are identical, even within the same ridge segment, because no two vent sites have exactly the same colonization history. The diversity of strategies for navigating this patchy, dynamic landscape, the different larval durations, the different dispersal distances, the different habitat specificities, is itself a product of millions of years of evolution in an environment where habitat availability has always been uncertain and distributed. The deep-sea organisms that have survived to the present are those whose dispersal biology has allowed them to track available habitats through geological time, to colonize new sites when they open, and to persist as a species even when individual habitat patches are lost. That dispersal biology, encoded in the developmental programs of larvae, shaped by the physics of deep ocean circulation, tested by the episodic creation and destruction of habitat patches over millions of years, is part of what makes the organisms of the deep sea extraordinary. They are not just adapted to extreme conditions. They are adapted to uncertainty, to impermanence, to the necessity of finding new places to live when the old ones disappear. That is a different kind of adaptation from the pressure tolerance and the membrane modifications, but it is equally essential to the deep-sea organism's success. And it is the kind of adaptation that, in a world where human activity is changing the distribution and availability of deep-sea habitats in ways that no previous geological event has produced, will be tested in ways that the evolutionary history of these organisms contains no preparation for. The organisms that are being tested most directly, the ones whose biology and ecology sit at the intersection of all these pressures, are the ones whose existence has raised the deepest questions about what life is capable of, and whose study continues to produce answers that the question of where life can exist anywhere in the universe cannot afford to ignore. The bioluminescent organisms of the deep ocean have solved a problem that no surface engineer has fully replicated: how to produce light with near-perfect efficiency using only chemistry without any of the thermal waste that makes every human light-producing technology a partial failure. The chemistry of bioluminescence, the reaction between a substrate molecule called luciferin and an enzyme called luciferase in the presence of oxygen, producing photons of light with almost no waste, heat has evolved independently at least 40 times in the history of life. Not 40 variations on a single ancestral light-producing system, but 40 entirely separate evolutionary origins, each starting from different chemical precursors, each arriving at a functional light-producing system through its own evolutionary path. This convergent evolution of bioluminescence, occurring independently across bacteria, dinoflagellates, jellyfish, squid, fish, and crustaceans, is one of the strongest arguments in biology for the power of natural selection to find similar solutions to similar problems from entirely different starting points. In the deep ocean below the photic zone, below the depth at which sunlight can penetrate, bioluminescence is not a specialized trick used by a few unusual organisms. It is a universal feature of the biological community. Estimates suggest that more than 70% of deep-sea organisms produce bioluminescent light of some kind, using it for some biological function. This prevalence makes the deep ocean, counterintuitively, a world not of absolute darkness, but of biological light. A world in which the primary illumination is not solar, but chemical, produced by living organisms and available only where those organisms choose to produce it. The darkness that a camera sees when it descends into the deep ocean, the darkness that is visible in every ROV video and submersible recording, is the darkness experienced by an organism that does not itself produce light, is not currently near an organism that does. For a bioluminescent organism in the deep, the environment is not uniformly dark. It is punctuated by flashes of light whose sources carry information about what produced them and why. The anglerfish lure is the most widely known example of bioluminescent predation, but it is far from the most sophisticated. The deep-sea dragonfish members of the family Stomiidae, found throughout the mesopelagic and bathypelagic zones, have evolved a bioluminescent capability that goes beyond simple light production. Several dragonfish species can produce far-red and near-infrared light, wavelengths that most deep-sea organisms cannot detect because their photoreceptors are tuned to the blue-green light that penetrates best from the surface. The dragonfish can see its own far-red light through photoreceptors that contain a specialized chlorophyll-derived visual pigment that is unique among vertebrates. A photoreceptor that has evolved specifically to detect the wavelengths that the fish itself produces. This creates a private channel. The dragonfish illuminates potential prey with light that the prey cannot see, detects the reflection from its photoreceptors that no other fish in the environment can use, and strikes at prey that is unaware it is being observed. It is a biological implementation of infrared night vision, evolved independently in a fish lineage without any access to the semiconductor physics that human engineers use to achieve the same function. The counter-illumination strategy used by many mesopelagic fish and squid is a different application of bioluminescence, not for predation, but for camouflage. An organism in the mesopelagic zone, at depths where dim light still filters down from above, casts a silhouette when viewed from below, a dark shadow against the faint background illumination from the surface. Predators hunting from below can detect this silhouette and use it to locate prey. Many mesopelagic organisms have evolved photophores, light-producing organs arranged along their ventral surface, producing bioluminescent light that matches the intensity and spectral composition of the downwelling light from above. From below, the illuminated ventral surface is nearly indistinguishable from the background illumination, eliminating the silhouette and making the organism effectively invisible to upward-looking predators. This counter-illumination camouflage is found in fish, squid, and crustaceans across the mesopelagic zone, representing another instance of convergent evolution, arriving at the same solution to the same predation problem from different starting lineages. The vampire squid Vampiroteuthis infernalis, whose Latin name means vampire squid from hell, is neither a true squid nor a true octopus, but a separate lineage of cephalopod that has been evolving in isolation in the deep ocean for hundreds of millions of years. It inhabits the oxygen minimum zone of the deep ocean, a layer at roughly 600 to 1,000 meters, where dissolved oxygen concentrations are so low that most organisms cannot survive. The vampire squid can survive there because of a hemocyanin, its blood's oxygen-carrying molecule that has an extraordinarily high affinity for oxygen, allowing it to extract usable oxygen from water that would suffocate other organisms. This gives it access to a refuge where predators cannot follow. Its bioluminescence is elaborate. It has photophores distributed across its body that it can activate in complex patterns, and it can produce a cloud of bioluminescent mucus, not ink as surface squid do, but glowing slime that confuses predators in the dark. Its arms are connected by a dark-colored cape-like webbing that it can wrap around itself to appear smaller or to conceal its photophores when it does not want to be visible. The vampire squid is not an aggressive predator. It feeds primarily on marine snow on the falling particles of organic matter using modified filaments to collect detrital material from the water. In the oxygen minimum zone, where food is scarce and predators are few, it has found a niche that no other cephalopod exploits, sustained by the slow rain of particles and protected by the chemical inhospitability of its environment. The siphonophores of the deep ocean represent perhaps the most extreme example of colonial organization in the animal kingdom. A siphonophore is not a single organism in the conventional sense. It is a colony of individual organisms called zooids, each of which is genetically identical to the others. They are all clones, produced by asexual budding from a single founding individual, but each of which has differentiated to perform a specific function within the colony. Some zooids are responsible for locomotion, contracting rhythmically to propel the colony through the water. Others are responsible for feeding, extending tentacles armed with stinging cells to capture prey. Others are responsible for reproduction, producing the eggs and sperm that allow the colony to reproduce sexually. Still others are responsible for flotation, containing gas-filled structures that regulate the colony's buoyancy. No individual zooid can survive on its own. Each is dependent on the others for functions it cannot perform. The colony as a whole behaves as a single integrated organism, coordinating the activities of its components through chemical and electrical signals, responding to the environment as a unit. And yet the colony is, at a cellular level, a collection of what began as separate individuals, a biological solution to the problem of multicellularity that is different from the solution found by every other animal lineage, in which the cells of a single individual differentiate into specialized cell types within a single body. The largest siphonophores can exceed 40 meters in length, longer than a blue whale, making them the longest animals on Earth, and they are found throughout the deep ocean, from the mesopelagic zone to the bathypelagic, trailing curtains of tentacles that can extend for meters in every direction and capture any organism small enough to be subdued by the nematocists of the feeding zooids. Dubia, a siphonophore found throughout the world's deep oceans, is thought to reach these extraordinary lengths regularly. Yet, despite their size, siphonophores are extraordinarily fragile. Their bodies are gelatinous and easily disrupted, and they cannot survive the pressure changes and physical stress of being brought to the surface in nets. Much of what is known about their natural behavior comes from ROV observations, which have revealed that siphonophores are far more abundant in the deep ocean than net-based sampling had suggested. Their transparent bodies, their slow drift through the water, and their tendency to remain motionless when not actively feeding make them difficult to detect except by cameras that approach slowly and carefully. The gelatinous organisms of the deep ocean, siphonophores, jellyfish, ctenophores, and salps collectively represent a biomass that is systematically underestimated by conventional deep-sea sampling methods, which tend to destroy delicate, gelatinous tissues. Recent acoustic and optical surveys, which can detect gelatinous organisms without physically capturing them, suggest that the gelatinous component of deep-sea biomass may be comparable to or greater than the hard-bodied component. If so, the energy flow through deep-sea food webs and the contribution of the deep ocean to global carbon cycling through the biological pump has been systematically underestimated because the most abundant organisms were the hardest to sample. The deep ocean's biological community is literally more than the sum of what has been caught in nets. A substantial fraction of its biomass, energy flow, and ecological function is embodied in organisms whose bodies dissolve before they can be brought to the surface and counted. The deep-sea fish that are visible in ROV footage, the snaggle-toothed dragonfish, the loosejaw, the bristlemouth fish of the genus Cyclothone, that may be the most abundant vertebrates on Earth by number of individuals, are not the charismatic megafauna that make deep-sea documentaries visually compelling. They are small, dark, and extraordinarily numerous, inhabiting the mesopelagic and bathypelagic zones in densities that dwarf the populations of any other vertebrate group. Cyclothone, the bristlemouth fish, is estimated to have a global population of quadrillions of individuals, making it almost certainly the most numerous vertebrate genus on Earth by individual count. It is roughly 3 to 5 centimeters long, nearly transparent, with a large mouth armed with tiny teeth used to capture small crustaceans and other zooplankton. It spends the day at depths of 200 to 1,000 meters and migrates toward the surface at night to feed in the richer waters of the upper ocean. One of millions of species that perform the diel vertical migration, the daily mass movement of organisms between the deep ocean and the surface that is one of the largest synchronized animal movements on Earth. The sheer biomass involved in this daily migration and the carbon that the migrating organisms transport from the productive surface to the deep during their daytime descent makes it one of the major pathways of carbon export. In the ocean, the obscure, rarely photographed, almost never discussed bristlemouth fish is, by the logic of numbers and ecology, one of the most important animals in the ocean. And it points towards something that the biology of deep-sea creatures, taken as a whole, consistently reveals. The most important organisms in an ecosystem are not always the most visible or the most dramatic. They are often the ones that no one is looking at. The DL vertical migration, the nightly ascent of mesopelagic organisms from the deep to the surface, and their return at dawn is a biological phenomenon of staggering scale. Every night, across every ocean on Earth, billions of tons of biomass move upward through the water column, feeding at the surface on the phytoplankton and zooplankton produced by photosynthesis, and then descend again at dawn, carrying with them the carbon they have incorporated from their nighttime feeding. When these organisms respire and excrete at depth during the day, they release carbon compounds below the surface layer, effectively transporting carbon from the sunlit surface to the deep in a process called active carbon export. The contribution of DL vertical migration to deep ocean carbon export is difficult to quantify. Precisely, the organisms involved are diverse, their migration distances variable, and their metabolic rates hard to measure in situ, but estimates suggest that active export by migrating organisms may contribute as much as 20 to 40% of total carbon export from the surface ocean, comparable to the passive sinking of particles that constitutes the better-known biological pump. The mesopelagic fish and crustaceans and jellyfish that perform this migration are, in effect, biological carbon pumps, living pipelines that transport the products of surface photosynthesis to the deep ocean every night, and have been doing so in one form or another for hundreds of millions of years. The acoustic signature of the deep scattering layer, the sonar reflection from the dense aggregations of gas-filled swim bladders in mesopelagic fish that appears to echo sounders as a false bottom in the ocean, was first described by naval sonar operators during World War II, who were confused by the anomalous reflection and initially suspected equipment malfunction. The daily movement of this false bottom, which ascends toward the surface at night and retreats to depth at dawn, tracking the movement of the organisms that produce it, was one of the first large-scale biological phenomena detected by acoustic instrumentation. The deep scattering layer is now understood to be one of the most abundant biological communities on Earth, a vast, globally distributed aggregation of organisms that human technology encountered acoustically decades before the organisms themselves were observed directly. It is a reminder that the deep ocean's biological community is not merely larger than what we have seen. In some cases, it was detected and quantified before it was ever observed, its presence inferred from the physics of sound reflection before a single specimen was photographed in its natural habitat. The relationship between the mesopelagic zone and the hadal zone, between the organisms that migrate daily and the organisms that live permanently at depth, is mediated by the biological pump that operates between them. The mesopelagic organisms consume surface production and export carbon to the deep through both active migration and passive sinking of their fecal pellets and dead bodies. That exported carbon is the primary food supply for the deeper communities, including ultimately the hadal zone. The dragonfish and the bristlemouth fish and the vertically migrating crustaceans of the mesopelagic are not merely interesting organisms in their own right. They are the connecting link between the sunlit world above and the permanent dark below the biological conveyor that translates photosynthetic production at the surface into the organic matter that sustains life at the bottom of the sun. The world's deepest trench. This connectivity means that changes in the mesopelagic zone, changes in the abundance, composition, or behavior of the organisms that perform the diel vertical migration propagate downward to affect the hadal communities. A warmer surface ocean that alters the composition of the phytoplankton community also alters the composition of the zooplankton that eat the phytoplankton, which alters the composition of the mesopelagic fish that eat the zooplankton, which alters the composition and quantity of the material that sinks to the deep. The hadal zone is insulated from the surface by 11 kilometers of water, but it is not independent of it. The connection runs through the bodies of millions of small organisms performing their nightly migration in the mesopelagic zone above, translating the chemistry of the surface into the chemistry of the deep through the universal medium of biology. The creatures of the deep sea are strange and extraordinary, and adapted to conditions that seem designed to prevent their existence. But they are not isolated from the world above them. They are part of it, connected to it through food chains and carbon cycles and evolutionary histories that span the full depth of the ocean and the full breadth of geological time. That connection is the next part of this story, what these creatures reveal about the limits of life, and what those limits mean for every question about whether life exists elsewhere in the universe. The question of whether life exists elsewhere in the universe, one of the most consequential questions in science, was transformed by the discovery of life in the deep ocean. Not gradually, not by accumulation, but suddenly, at a specific moment. The moment in February 1977, when the crew of the submersible Alvin descended to the Galapagos Rift and found an entire ecosystem thriving in complete darkness, powered not by sunlight, but by the chemistry of hydrothermal vents. Before that moment, the search for extraterrestrial life was organized around a single assumption: that life requires sunlight. The habitable zone around a star, the range of orbital distances where liquid water can exist and life might therefore be possible, was defined by the requirement for solar radiation to drive photosynthesis. The assumption was so fundamental that it was rarely stated explicitly. It was simply the framework within which the question was asked. The vent ecosystems demolished that assumption. If life could exist in the complete absence of sunlight, driven entirely by chemosynthesis, then the habitable zone was not defined by the distance from a star at which photosynthesis was possible. It was defined by the availability of liquid water and chemical energy conditions that could be met in environments far beyond what stellar radiation could reach. The implications cascaded immediately through astrobiology. Europa, one of Jupiter's moons, was already known to have a subsurface liquid ocean, kept liquid by the tidal heating generated by Jupiter's immense gravitational field. If hydrothermal vents existed on the floor of Europa's ocean, if the same geological processes that drive vent activity on Earth were occurring beneath kilometers of European ice, then the conditions for chemosynthesis-based life would be present, entirely independent of the Sun. The distance from Jupiter to the Sun, which had previously seemed to place Europa comfortably outside any habitable zone, became irrelevant to the question of whether life could exist there. Enceladus, a moon of Saturn, subsequently provided even more compelling evidence. The Cassini spacecraft, in orbit around Saturn from 2004 to 2017, discovered that Enceladus was venting water vapor and organic compounds from its south pole through cracks in its icy surface material, erupting from a subsurface liquid ocean through geological activity that bore striking resemblance to the processes driving hydrothermal vent activity on Earth. The organic compounds detected in Enceladus's plumes included hydrogen gas, a product of the reaction between hot water and rock that also fuels chemosynthesis at Earth's hydrothermal vents, and complex organic molecules whose presence in the plume suggested active chemistry in the subsurface ocean. Enceladus has liquid water. It has hydrothermal activity. It has organic chemistry. Everything that Earth's vent ecosystems need is apparently present in the ocean beneath the ice of a moon of Saturn. Whether life has actually arisen there is unknown. But the framework for asking the question, which the deep-sea discoveries made possible, is now well established. The deep-sea creatures whose biology has informed this reassessment of habitability are not merely existence proofs, demonstrations that life can exist under conditions once considered incompatible with it. They are, more specifically, demonstrations of the mechanisms by which life adapts to those conditions. The TMAO chemistry of the snailfish, the membrane lipid modifications of the amphipods, the endosymbiotic partnerships of osedax and the vent tube worms, the chemosynthetic metabolism of the vent bacteria. Each of these is a specific molecular or biochemical solution to a specific physical or chemical challenge. And each of these solutions, or variants of it, might be available to life in any environment where similar challenges exist. Life on Europa, if it exists, faces high pressure, cold, darkness, and the need to extract energy from chemistry rather than light. The deep-sea organisms of Earth have solved each of those problems through mechanisms that are, in principle, available to any carbon-based biochemistry. They are not uniquely earthly solutions. They are solutions that the laws of physics and chemistry make available to any life that faces similar challenges, regardless of the star it orbits or the ocean it inhabits. This universality of the solutions found by deep-sea life is part of what makes the deep ocean so scientifically important to astrobiology, more important, arguably, than the headline discoveries of extreme temperatures or pressures. The headline discoveries establish that life can exist in extreme conditions. The biochemical details establish how, and the how is transferable in a way that the headline is not. Understanding the specific molecular mechanisms of pressure adaptation in hadal organisms gives astrobiologists a prediction. If life exists in the high-pressure ocean of Europa, it probably uses pressure-stabilizing small molecules that function like TMAO. If life exists in the chemically reduced environment of Enceladus's hydrothermal systems, it probably uses chemosynthetic metabolism that resembles in its basic logic the metabolism of Earth's vent bacteria. The deep ocean is not just a place where life has adapted to extreme conditions. It is a library of adaptive strategies, a collection of tested biochemical solutions to the challenges of extreme environments that can inform the search for life in any extreme environment anywhere. The organisms at the very bottom of the Mariana Trench, the amphipods at the Challenger Deep, the foraminifera in the sediment, the bacteria processing organic matter at temperatures near freezing under pressures exceeding 1,000 atmospheres, are among the most extreme practitioners of life on Earth. But they are not at the limit of what life is capable of. The hyperthermophilic archaea of hydrothermal vents can grow at temperatures exceeding 120 degrees Celsius, sustained under the high pressure of the vent environment, which keeps water liquid at temperatures far above its surface boiling point. The halophilic bacteria of deep brine pools can function in salt concentrations that would desiccate any cell not specifically adapted for them. The radioresistant bacterium Deinococcus radiodurans can survive radiation doses that would destroy the DNA of any other known organism using a DNA repair system of extraordinary efficiency. The tardigrades, microscopic animals found throughout the ocean and on land, can survive vacuum, radiation, desiccation, and temperature extremes that would kill every other animal. Life, as practiced on Earth, does not have a single limit. It has many limits, each specific to a particular type of challenge, and each of those limits has been pushed further by the organisms that have had the longest time and the strongest selection pressure to push it. The deep-sea organisms are specialists in a specific set of challenges: pressure, cold, darkness, chemical scarcity. In those challenges, they are the most extreme practitioners known. But the broader catalog of extreme environment life on Earth suggests that the space of conditions in which life can exist is far larger than the space in which human beings can live or even survive, and that the broad principles governing that space are universal enough to apply across planetary bodies that have never shared an evolutionary history or a biochemical ancestry with Earth. The discovery of life in the deep ocean did not merely expand the known range of habitable environments. It changed the conceptual framework within which habitability is understood from a question about proximity to stars and access to sunlight, to a question about the availability of liquid water, chemical energy, and time. Time, in this context, is not incidental. The organisms of the deep ocean are not recent arrivals. The hadal amphipods descend from lineages that colonized the deep ocean tens of millions of years ago. The chemosynthetic bacteria of hydrothermal vents have been present at vent systems for hundreds of millions of years. The deep ocean microbial communities whose metabolic activities drive the global carbon and nitrogen cycles have been operating in some form for billions of years. Life at depth is ancient life, refined by evolutionary pressure over timescales that dwarf the duration of any human civilization. Its biochemistry is not a rough draft. It is a final product tested, refined, and optimized by more generations of selection than can be meaningfully imagined. If life exists elsewhere in the universe, it has had comparable time. The universe is 14 billion years old. The conditions for liquid water and chemical energy have existed in various places throughout most of that time. Life that has had billions of years to adapt to the specific challenges of its environment may be as refined in its own biochemistry as the organisms at the bottom of the Mariana Trench are in theirs. The question is not whether such life could exist. The question is where it is, and how we would recognize it if we found it. The deep-sea organisms of Earth provide the clearest answer available to that question. They are the existence proof that life adapts, that it finds ways, that it persists in conditions that seem designed to prevent it. They are the demonstration that the solutions life finds to extreme challenges are not arbitrary, but are constrained by the same chemistry and physics that operate everywhere in the universe. And they are, in their collective strangeness and their collective abundance and their collective ecological importance, a reminder that the definition of life, what it requires, what it can tolerate, what it can produce, is not written by the organisms visible on the sunlit surface of the one planet where we have thus far looked. It is written in the full diversity of the living world, from the surface to the bottom of the deepest trench, from the warmest vent to the coldest sediment, from the largest whale to the smallest virus. Everything that lives is part of the definition, and most of what lives, by biomass and by metabolic activity and by ecological function, lives in the dark. The census of deep-sea life is incomplete in ways that have direct consequences for how seriously the deep ocean's role in global ecology is taken. When policy discussions about ocean conservation focus on coral reefs and fisheries and the sunlit coastal waters that are most directly accessible to human observation and human activity, they are discussing a small fraction of the ocean's total biological diversity and ecological function. The deep ocean, which constitutes more than 95% of the ocean's habitable volume, is represented in those discussions primarily by its absence from the data. It is not that the deep ocean is known to be unimportant. It is that the deep ocean is incompletely known, and incompletely known things tend to be treated as unimportant. The organisms whose existence has been documented in the deep sea, the snailfish and the amphipods and the zombie worms and the vampire squid and the bristlemouth fish and the siphonophores and the chemosynthetic bacteria of the vents, are the ones that have been found. The ones that have not yet been found, which likely exceed them in diversity and may rival them in ecological importance, are still waiting to be discovered. The pace of deep-sea discovery has been accelerating as the technology improves. Each new generation of ROVs and AUVs is more capable than the last, able to reach greater depths, carry more sophisticated sensors, spend more time at depth, and return higher quality data. The genetic sequencing technologies that allow researchers to characterize microbial communities from sediment samples have become faster and cheaper by orders of magnitude in the past decade. The computational tools that process the resulting data are improving rapidly. The deep-sea biology of 2025 is doing things that the deep-sea biology of 2000 could not have imagined, and the deep-sea biology of 2040 will do things that the deep-sea biology of 2025 cannot yet envision. Every expedition brings back new species, new behaviors, new biochemistries, new ecological relationships. The deep ocean is not a solved problem. It is a frontier, and the frontier is moving. The organisms at the frontier, the ones that have not yet been described, not yet been named, not yet had their biochemistry analyzed, or their ecological role understood, are doing what they have always done, regardless of whether human science has reached them. They are eating and being eaten, reproducing, adapting, dying, and being replaced by their descendants in the dark and the cold and the pressure, in the most remote environment on the surface of this planet. They have been doing this for longer than any human institution has existed, longer than any human language has been spoken, longer than any human thought has been recorded. They will continue to do it long after the current moment of accelerating discovery has passed, whether that discovery continues or is interrupted by the competing demands of a civilization with limited attention and limited resources. What the discovery of these organisms has given us is not merely a list of species or a catalog of adaptations. It has given us a revised understanding of what life is, what it requires, what it can tolerate, what it produces, and where it might be found. That revised understanding is one of the most consequential scientific developments of the past 50 years, and it came directly from looking at the creatures of the deep ocean. From the moment in 1977, when the Alvin's cameras first illuminated the improbable life around a hydrothermal vent, to the ongoing expeditions that continue to find new organisms in the deepest trenches and the most extreme habitats that this planet offers. The creatures that should not exist have told us more about the nature of existence than almost anything else we have found. The specific creatures whose biology has driven these revisions, the snailfish at the edge of the vertebrate depth limit, the zombie worm-farming bacteria in whalebone, the vampire squid in its oxygen refuge, the siphonophore trailing 40 meters of gelatinous body through the bathypelagic dark, the bristlemouth fish present in quadrillions are each a different facet of the same truth. That truth is not simply that life is resilient, or that life is adaptable, or that life finds a way, though all of those things are true. The deeper truth is that life, wherever it has time and selection pressure and the raw materials of chemistry, tends toward complexity. Not inevitably, not universally, not in every lineage, at every moment, but as a statistical tendency across the full space of evolutionary possibility. The deep ocean has had time, hundreds of millions of years at depth and selection pressure. The unrelenting physical demands of pressure and cold and darkness and the raw materials of chemistry, the organic. Compounds raining down from above, the inorganic minerals of the trench walls, the reduced chemicals seeping from the geological activity below. From these inputs, evolution has assembled a community that the cameras of the ROVs have been finding, piece by piece, expedition by expedition, over the past few decades. That community is complex. It is ecologically structured. It is biochemically sophisticated. It is, by any reasonable measure, remarkable. And it occupies less than 0.5% of the ocean floor in the most extreme physical environment that the surface of this planet contains. What occupies the remaining 99.5% of the ocean floor in the somewhat less extreme conditions of the abyssal plain is a question that has barely been asked with the systematic rigor it deserves. The abyssal plain is the largest single habitat on Earth, a vast, flat, cold, dark expanse at depths of 3 to 6 kilometers, covering more than half of the planet's surface. Its biological communities are poorly characterized, its microbial diversity largely unexplored, its ecological dynamics understood only in outline. The hadal zone gets more attention partly because it is more extreme and therefore more dramatic, partly because the few expeditions that have reached it have returned with sufficiently remarkable results to justify the investment. But the organisms of the abyssal plain, less dramatic, in less extreme conditions, but covering an area that dwarfs every other biome on Earth, are equally unknown and potentially equally important to the ecological and chemical functioning of the global ocean. The next chapter of deep-sea discovery may not be written in the trenches. It may be written on the plains. The ocean's deepest creatures have been patient. They have been waiting in the dark, under the weight of the water above, for the attention of the species that has been looking at the stars while the most biologically complex environment on this planet remained unobserved below the surface of the world's oceans. That attention is arriving, slowly and unevenly, and with insufficient funding and insufficient urgency, but arriving. Each expedition that descends finds something unexpected. Each unexpected find revises something that was previously considered settled. The process is cumulative and self-reinforcing. Each discovery makes the case for further exploration more compelling, because each discovery demonstrates that what has already been found is a small fraction of what remains to find. The deep ocean is not running out of surprises. It has not come close to running out of surprises. It will not run out of surprises within any time frame that human science can currently imagine. If this journey moved something in you, consider subscribing to Sleep on Deep. Every week, we descend a little further. The ocean is waiting.